A very good afternoon to everyone present here. I'm very pleased to introduce our next speaker for the session. We have among us Dr. Tulika Prasad, who is the Assistant Professor, AIRF and Center for Nanosciences, Jawaharlal Nehru University. Her specialization includes nanomedicine, infectious disease biology, and multi-drug resistance. She has received the postdoctoral fellowship from DBT and CSIR and worked at the Special Center for Molecular Medicine, JNU. She is also a recipient of NIH Visiting Fellowship at Liver Diseases Branch, NIDDK, NIH USA for the year 2008 to 9. Tulika ma'am is also a recipient of this several Young Scientist Award, Ranbaxy Young Scientist Scholar Award for 2008 in the field of medical sciences by Ranbaxy Science Foundation, Innovative Young Biotechnologist Award 2008 by Department of Biotechnology, Government of India. She also received the Best Hall Presentation Award in Young Scientist Conclave at Indian International Science Festival 2016 by the Ministry of Science and Technology and Ministry of Earth Sciences, Government of India. She is currently leading a research team of 8 students. We are delighted to have you here, ma'am. Now I'd like to invite you to take over the session on functional nanostructured materials in nanotechnology and nanomedicine. Over to you, ma'am.
Thank you, Ritika, for a nice introduction. Good afternoon, everyone. It's my pleasure to present my work here. I'll just share my screen. My screen is visible to everyone?
Yes, ma'am, it's visible.
First of all, I would like to thank the organizers and of this conference and Dr. Asmita Das for this opportunity so that we all have gathered, and it was lovely hearing all the talks, wonderful talks, and an opportunity to present the work here. So I would be talking on functional nanostructured materials for applications in nanobiotechnology and nanomedicine. So many of you of the speakers already are from JNU, but still for the students, I would like to share. So this is our campus. So this is our central library. This is our convention center, and this is an aerial view of JNU, and this is our administration. And you find a lot of wild animals also in JNU. So you find peacock, nilgai, and a lot of birds and butterflies, and a lot of greenery also within the campus. Now coming to my department, so I belong to 2 departments. So Special Center for Nanoscience, where I teach, and my MTech students and PhD students are there. And this center has 2 programs, MTech and PhD programs we conduct in nanoscience, and it's an interdisciplinary academic and scientific research program. And this is the other place, other department, AIRF. So this is the building, how it looks like. And this is the walkthrough, the building. And this offers training, workshops, seminars, conferences for the JNU masters and PhD students. And it also provides the research infrastructure to various researchers. And it has a lot of infrastructural facilities, which we all from JNU can use. We have the transmission electron microscopy and the scanning electron microscopy, lot of XRF facilities and XRD X-ray based instrument facilities, lots of fluorescence based facilities. Then you can do a lot of ultrastructure and surface morphology studies. You can do a lot of live imaging and time-lapse imaging, confocal, and you can use different techniques for the studying different biological interactions, and you have also the mass spectrometry facilities. Now, this is my research laboratory. So primarily I'm working on the novel determinants of MDR in microbes, and nanomedicine for infectious diseases, and lipid biology and role of lipids in mediating drug resistance. So the 2 organisms that I'm working primarily on is Candida albicans, which is an opportunistic fungal pathogen, and Mycobacterium tuberculosis, which is a pathogenic bacteria. Now, this is my research group. So this is Radhakrishnan, whose work I will be presenting today. So he finished his PhD last year and is now working in NCDC, National Center for Disease Control, New Delhi. And she's Richa Singh. She worked on the molecular lipid-mediated drug resistance mechanisms in Mycobacterium, and she submitted this year. And this is Arvind Kumar. He also submitted this year. And he worked on the bioequivalence evaluation of drugs in human plasma. And this is Preeti. She is in her final year of her PhD, and she's working on zinc oxide quantum dots, so part of her work. And she also worked on DNA, nucleic acid based DNA dots and DNA-based hydrogels. So I would be presenting part of her work. And Anam Reis, whose oral talk was day before yesterday, she works on carbon-based quantum dots. So part of the work I would be present. So these 4 nano materials I would be discussing today. And she is Megha, so she is in her second year. She is working also on nucleic acid-based dots. And these are 2 MTech students, Saurav and Fareen. So they are working on 2 different materials, all biocompatible, non-toxic nanodots. And he's the laboratory staff without whom the lab would not run properly. And these 2 are students who are about to join the lab. So now coming to the work. So first I would like to introduce a little bit about infectious diseases. So according to World Health Organization, the infectious diseases are caused by the pathogenic microbes such as the bacteria, fungi, viruses or parasites, and it can be spread directly or indirectly from one person to another. And zoonotic diseases are also the infectious diseases which are spread from animals to humans. So the infection can be caused in the 4 steps. One is, first is the invasion of the body of an organism by disease-causing agents. Then is the multiplication within the body, then reaction of the host tissues towards the infectious agents, and then finally the toxins are produced which are harmful to the body. Now, there are many common infectious diseases, of which some are the viral diseases, some are the bacterial, some are the parasitic diseases, some are the fungal diseases, and these are the different causative agents. So of which we work on the Candida albicans and some other Candida species also, which causes the candidiasis, and Mycobacterium tuberculosis, which is responsible for causing tuberculosis. Now, what are the challenges in infectious diseases? So it is the ability of the microorganisms to evade the chemotherapy, and they keep on continuously evolving the strategies for inactivating most of our potent antibiotics. And there are continuously newly emerging infectious organisms. One recent example is the novel coronavirus. Then there is a global spread of drug-resistant pathogens. So pathogens are continuously emerging, developing new resistance mechanisms. And then there is the threat of bioterrorism also. Now, how can we overcome? So there is one part that is the basic science. So we can have a deeper understanding of the microbial physiology and comprehensive understanding of antibiotic resistance. So these 2 can together help in designing new strategies which can help us to control these infectious diseases. And second is the technology of anti-infective chemotherapy, which is a renewed commitment to the discovery of new antimicrobial molecules and therapies which can help us also to control these infectious diseases. Now, as I talked about drug resistance, so what is drug resistance? So it is actually the failure of therapy due to emergence of resistance to one or more drugs. And when the resistance is towards a group of unrelated compounds, then that cross resistance is called multi-drug resistance. Now, how this multi-drug resistance evolves? So as you can see, there are primarily many mechanisms by which the drug resistance may evolve. Like there may be, the target may be altered so that the drugs are not able to identify the target, and it may be by point mutations, overexpression of the drug targets, gene amplification, gene conversion. Then maybe the drug extrusion, Now, because of the increased efflux of drugs by overexpression of the drug efflux pumps, so there are drug efflux pumps in proteins in the cells which effluxes out anything from the cells which is not required by the cells. Third is there may be some alterations in the cell membrane, primarily in the lipids, which may cause permeability constraints due to which the molecules will not be able to enter inside the cells. And lastly, there would be drug inactivation. There may be modification and degradation of the drugs so that the drugs become inactivated, like the beta lactamase producing bacteria. They often cleave the beta lactam rings of the antibiotics, and that is how they inactivate the drugs. Now, as you see in this animated image, there are the drug molecules, and these are the drug efflux pumps, which are effluxing out the drugs. And here you can see properly, these are the efflux pump proteins. So there are porins and some other channels through which the drug molecules enter the cells. But as soon as they enter the cell realizes they don't want the drug molecules there because they are not liking it. It is causing harm to them. So immediately they will efflux out the drug molecules. So this is primarily a very important efflux mechanism, not only found in the microbes, but also frequently found in the cancer cells. So now, what are the recommendations on resistance to respond to issues of resistance to antimicrobials? You can establish a simple and readily searchable antibiotic resistance database in which the participating institutions can upload the resistance data in real time. You can have new diagnostics to detect the pathogens and their resistance to inform therapy in real time. You can develop strategies which can direct the pathogenic organisms targeted and avoid targeting the host or other beneficial organisms. And you can identify the mechanisms, their evolution, and that is how you can develop new strategies that target and selectively block the antibiotic resistance mechanisms. And you can also explore the combination therapy in combating the resistance, which is a very good way that you can try more than one drug in combination to combat the resistance, which we were discussing in the morning session also. So now there is need for development of novel antimicrobials. Now there has been less development of antimicrobials and more attention has been given to other areas to develop more drugs. So the microbial pathogens have got little less attention, and there has been less innovation as far as the antibiotics are concerned. Like the recent, one of the recent ones is in 2000, the linozet, which was discovered in 2000. And in the last 50 years, the medicinal chemists have been able to reshape the scaffold of earlier antibiotics, both natural and synthetic. And some of these include the beta-lactams and third generation of macrolides. Now, example of some combination therapy, as I told, amoxicillin is a well-known antibiotic used to treat bacterial infection. But in these days, you will find it is in combination with clavulanate potassium, which is also known as the augmentin. So this actually attacks the beta-lactamase enzyme of the bacteria, and in combination, the bacteria then no longer can deactivate the amoxicillin, and as a result, this becomes an effective combination. Then combination therapy has also been found to inhibit the resistance, which has been used in the treatment of HIV, like heart therapy, and tuberculosis, where you have a combination of drugs like isoniazid, rifampicin, pyrazinamide, streptomycin. Now, coming to different technologies, how to develop drugs or how to combat this drug resistance. So there are some different branches of technology like biotechnology, nanotechnology, which combinedly give the nanobiotechnology, life sciences, nanoarrays, biomarkers, cell therapy, and all these together, nanobiopharmaceuticals, drug delivery, drug discovery, molecular diagnostics. So they are all interrelated, and they have given to the development of a new branch called nanomedicine, and which can in future lead to the development of personalized medicine. Of course, we look forward to it, although it is still a long way to go and reach there. So now, what is nanotechnology? So anything you design or manipulate the materials within a scale of 1 to 100 nanometer, that branch is nanotechnology. And nanotechnology has attracted immense attention in the recent years and showed potential in areas of societal importance. Now there are engineered nanoparticles like metal nanoparticles, gold, silver, copper, metal oxide, zinc oxide, titanium oxide, silicon dioxide, cerium oxide, quantum dots, carbon nanotubes, graphene, fullerene. Now, as you see, you can tune. The advantage of using nanotechnology is that you can tune the optical and the chemical properties of your nanostructures and design it the way you want it for your application. Now, the interdisciplinary field with applications which combines the applications of nanotechnology with biology is called nanobiotechnology, and it is an interdisciplinary science which involves all the three areas: physics, chemistry, and biology. And nanobiotechnology is associated to both physicochemical attributes of nanostructured materials as well as the various aspects of molecular biology and immunology. Now, biomolecules such as proteins, nucleic acids, supramolecular complexes are used for the fabrication of nanostructures from inorganic and organic nanoparticles and vice versa. And then they can be used for further medical and biological applications. Although significant challenges still exist and it is still in the infancy, integration of nanostructured materials with biology has led to the development of nanomedicine, which holds immense promise in revolutionizing the healthcare. So now, as you see, nanomaterials can be used for targeted therapy, diagnostics and imaging. They can be used for theranostics, where you can have both the diagnosis and treatment. And so nanomedicine can have multiple applications, and it can be also for tissue engineering, bone engineering, and different therapeutic and diagnostic applications. Now, there has been different functionalized nanostructures which have been used for as analytical tools, therapy, contrast agents, diagnostic devices, vaccine delivery vehicles, and theranostics. So now the nanoparticles may be synthesized nanoparticles, or they may be the engineered nanoparticles. So they can be inorganic or organic nanoparticles like liposome, polymeric micelles, or they can be inorganic like gold, silver, copper, silicon dioxide, titanium dioxide. And as far as applications in infectious diseases are concerned, they can be used as drug delivery system, or they can be used as antimicrobial agents. Now, in nanomedicine products, a lot of work has been done in the therapeutic area, as you can see in this pie diagram, and little bit in regenerative, in vaccines, little still less, then in vivo diagnostics, and in vitro diagnostics, less amount of work has been done. Now, as far as among the diseases, the maximum amount of work that has been done in the nanomedicine is for the cancer disease, followed by this one, that is the central nervous system products. Then rest is for anti-infective or others, or anti-inflammators that has been done in the area of nanomedicine. Now, it takes a lot of work, and the nanomedicine area is yet to pick up, because you have to first synthesize the nanostructures, characterize, do all the studies, then the cytotoxicity studies, analyze the mode of action, preclinical studies, then clinical trials, then only you get the drug administrative approval as per the different countries. So it's a long process. Any nanomedicine would take to develop as a drug. But still, there are a lot of anti-cancer drugs available in the market. So new nanostructures with multiple biomedical applications are gathering interest, and they have been attached to prodrugs, drugs, tracking entities, targeting molecules, and they are all under continuous investigation. So I would be covering little bit on these four nanostructures. So one would be the zinc oxide quantum dot. So we prepared it, and the size was 5 to 6 nanometer. Nanosilver of 20 nanometer size. Then water-soluble, biocompatible, multifunctional DNA dots, which were of 4 to 5 nanometers, and DNA dots-based fluorescent hydrogels for drug delivery and biosensing. And the fourth material would be water-soluble, non-toxic fluorescent carbon dots of 2 nanometer size. Now, first we come to zinc oxide carbon quantum dots. So we published 2 papers this year, and this is Preeti's work that I would be discussing. So why we selected zinc oxide? So it is a non-toxic nano compound as compared to other competitive nanomaterials, and it is widely used in the sunscreen and cosmetic industry. And the advantage is that they have a large surface area relative to their size and high catalytic activity. And the exact physical and chemical properties you can tune based on the method that you are using for your synthesis. So we synthesized the zinc oxide quantum dots using the wet chemical route. And as you see, these were the different characterization, physicochemical characterization techniques that we used. So we found absorption maxima, UV visible absorption maxima at 365 nanometer, which correspond to enhanced band gap of 3.4 electron volt, which shows that it is due to the smaller size of zinc oxide quantum dots. Then using the XRD, we confirmed that they have single phase hexagonal woodsite structure, and the size was found to be as between 5 to 6 nanometer. We confirmed it using other techniques. So we used the scanning electron microscope, which revealed the, spherical zinc oxide quantum dots with smooth surface, and the EDX energy dispersive spectra. It showed that the elements present are only zinc and oxygen. That means the material that has been synthesized is pure. And transmission electron microscopy further confirmed the size as 5 to 6 nanometer, and the lattice structure was determined using HRTEM, high resolution TEM, and we confirmed that the lattice spacing, d-spacing is 0.255 nanometer, which corresponds to that of the zinc oxide. So using the HRTEM, we found the SAID pattern, selected area electron diffraction pattern, which confirmed the results of the XRD. And again, the SEM EDS also confirmed what we found from the SEM EDS, that it consists only of the zinc and oxygen, and indicating no trace of impurity in the sample. Now, we checked it against 2 microbial pathogens. So one was E. coli and the other was C. albicans, and we found there was dose-dependent toxicity. So as you see, the optical density of the cells are reducing. That means the growth of the cells are affected, and what you see here is the percentage of infection inhibition. So as you increase the concentration of the zinc oxide quantum dot. So here you see almost 71% of the cells getting inhibited, and here 91% of the cells getting inhibited. It showed better action against the Candida albicans cells. Now for clinical study, clinical strains, we confirmed using some ESBL, which is enhanced spectrum beta lactamase producing MDR isolates of E. coli, which we got from a clinic. From a pathodiagnostic lab, and these strains were showing resistance to the penicillin derivatives penams, cefems, monobactams, carbapenems, and carbabacifems. So, and these strains we found that, so this is the resistance profile that we worked upon in collaboration with the pathodiagnostic lab. And as you see, most of all the strains, the 4 strains that we used, are showing resistance towards all the 3rd and 4th generation of antibiotics that are commonly used today. So as you see here, so this is the spot assay. So this is the control where you don't use any drugs. So you see, this is the serial dilution of the cells, and you see the cells are growing properly. And as you dilute the cells, you see less cells growing. And here you are increasing the concentration of the zinc oxide quantum dots, and you find all the cells are becoming susceptible, and there is thin papery kind of growth found for the cells. Similarly, for Candida albicans, we used isogenic pair of isolates, which means the same. They are collected from 3 different AIDS patients, and they were kind gift from Professor Joachim Moschasser from Würzburg University, Germany. And so these isolates were collected over a period of more than a year, and the same strain over a period of time became resistant and was expressing all the resistant mechanisms. So as you see, this G1 is, G5 is derived from G1, F5 is derived from F1, and GU5 is derived from GU4. And these are much resistant isolates where you find the MIC of the drugs very high. And we could find that zinc oxide quantum dots at the MIC concentration of 200 microgram per ml, you find that the cells are actually getting susceptible as compared to the control. So we could conclude that they were able to counter the MDR in both the bacterial and the fungal isolates, and there was killing of the microbial cells, the clinical resistant microbial cells. Now, due to limited antifungal arsenal, researchers have sought to improve the treatment via various approaches, and synergistic approach is a very good approach used by the combination of antifungals, and which can reduce the dosage of individual antifungals, individual drugs, and it can contribute to reducing the toxicity and could serve as an alternative for treatment. So as you see here, so first I'll show that we for all these clinical isolates, we did dose-dependent toxicity analysis, and we found that zinc oxide quantum dots exhibited dose-dependent antifungal toxicity. As you see, the absorbance of the cells or the optical density of the cells are reducing with increase in the dose. So this is 25. Then this is no zinc oxide quantum dots. Then this is 25, 50, 100, 200. And this is the percentage of growth inhibition. As you can see, almost more than 80% growth inhibition is obtained for all the isolates. Now we confirm this using the synergy checkerboard assay. So here you use the serially increasing concentrations of 2 compounds. So this is compound A, this is the second compound, and then this is the where you find the synergy is happening. So synergy, how it is calculated? So synergy is calculated as A is the MIC in combination of first drug by MIC of the first drug alone. B is the MIC in combination, and MICB is combination alone. So this will give FIC fractional inhibitory concentration for a first drug, and this will give for the second drug, and together they will give the FIC index. Now, if FIC index is less than 0.5, it is synergy. If it is more than 4, then it is antagonism. That means one drug is acting antagonistic to the other drug. And additive or indifference when both the drugs do not make any difference to each other. Now we come back here. Now, when they were used in combination with different drugs, like we used fluconazole and alone. So this is fluconazole alone, and this is fluconazole plus zinc oxide. So you can see here that normally the MIC concentration of zinc oxide is 200. Here we use 25 microgram per ml only. And you can see the percentage of growth inhibition. It has increased for all the cells. Similarly for ketoconazole, so it varies from drug to drug. So we use 2 azole drugs, fluconazole, ketoconazole, and 2 polyene drugs, amphotericin B and nystatin. So both are type of antifungal drugs. So as you see here, so I come straight away here, as you see in combination, it works very well. So, and we calculate the value, and found for all the drug combinations, it was well below 0.5. So it was showing synergism with all. Now to find out whether 25 microgram per ml of zinc oxide quantum dots is biosafe or not, we found that it almost 93% and 86% cell viability of HeLa cells were observed in presence of 5 microgram per ml zinc oxide quantum dots and 25 microgram per ml quantum dots concentration. So to conclude for this part, the synergism between zinc oxide quantum dots and individual antifungal drugs were observed for all drug susceptible and drug resistant clinical isolates of Candida albicans. So probably this can be used as a potential adjuvant in combination antifungal therapy against MDR fungal pathogens, wherein we can reduce the drug toxicity, and their simultaneous multi-targeted synergistic action can also limit the development of fungal drug resistance. Now coming to the silver nanoparticles. So this is Radhakrishnan's work. So two papers were published in 2018 and the other two papers are under communication. So I skip these slides. So if you see, this is how a fungus cell undergoes budding, and one cell can keep growing into multiple cells. And Candida is an opportunistic pathogen, and it can form the bud form and both the hyphae form. So this is the more virulent form, and it can exist in both the forms. So there are various drugs used now. Why silver? So silver is a common disinfectant used in the treatment of clinical diseases, eye prophylaxis, and a lot of other diseases also. And polystyrene and human epithelial cells pre-treated with the nanosilver is already found to significantly reduce the adhesion of biofilms. And it is less toxic than others and more toxic towards microorganism, and it is also used in many creams, solutions, lotions, and different biological catheters as a coating. So now we wanted to find out that how the silver nanoparticles are acting on the microbial cells. So we could make the nanoparticles of 20 nanometers by using the chemical reduction method, and we characterized different methods, and we found that it contained 98% pure silver. And it showed the dose-dependent antifungal activity, as you can see here. So for, compared to 5 microgram per ml, 40 microgram is killing more cells, and you can see here the growth of the cells is also reducing. Now, how they kill the cells? So we found that it generates the reactive oxygen species, and it was also dose dependent. So we increase the silver nanoparticles, and we find the reactive oxygen species generation is augmented. And we use this DCFTA dye, which within the cell, it gets, if it reacts with the reactive oxygen species within the cells, it forms DCF, which is dichlorodifluorescein, which is a green fluorescent dye, and it gives this color, and which shows that what is the percentage of the reactive oxygen species within the cells, as you see. And when we treated it with an antioxidant ascorbic acid, we found there is reduction in the ROS because it detoxified the ROS generated within the cells. Now, interestingly, what we did, we added antioxidant to the silver nanoparticle treated cells, and we found that there is restoration of the antioxidant there is restoration of the ROS levels to the basal level where no AGNP was added, but the cell was still getting killed and it was not getting restored to as the control level. So the AGNP induced cell toxicity of the Candida cells could not be completely reversed. So there must be other mechanisms also which are coordinately acting along with the generation of the reactive oxygen species, and we found that there are a lot of cellular changes that are happening. So there are a lot of alterations in the ultrastructure. So we found that the cell wall and cell membrane of the cells are compromised. And there are also changes in the ergosterol, which is an important membrane sterol, which maintains the cellular integrity. And there is also changes in the level of the fatty acids, especially the oleic acid, which is an important signaling molecule which plays an important role in the pathogenicity of the Candida albicans. So we found with the treatment of the silver nanoparticles, it is significantly reduced. So these two are important fatty acids. Both are unsaturated fatty acids which were reduced, and probably that may be possible that it is responsible for reducing the pathogenicity and also reducing the hyphal morphogenesis. So there were different other cellular changes also. So we found that the fluidity of the changes and the organizational dynamics of the interior of the membrane is changing. Hydrophobic interior of the membrane is changing. So as you can see, uh, so there are different fluorescent probes. So we used DPH, which goes and enters the hydrophobic core pockets, and how easily it enters will give you a measure of the fluidity. If it is going inside the cells very easily, that means the cell is fluid. And if it takes some time to enter into the cell, that means it is rigid. And we also checked how it affects the drug efflux pumps. And interestingly, we found that the drug efflux pumps are significantly downregulated in presence of the silver nanoparticles. So as you see here, that a normal candida cells can form hyphae like this, whereas the silver nanoparticles treated cells could not form hyphae even after incubation of the nano cells with nanoparticles for the same time as the normal cells. So you can just see this. So this is the hyphal formation, as I mentioned in the slide, in the beginning of the slide, that they are dimorphic. They can exist in the cell form, bud form, and also in the hyphal form. So to conclude, AGNP augmented ROS generation. It altered the cellular ultrastructure, cell surface morphology. It reduced the ergosterol and altered the fatty acid composition. It altered the membrane fluidity and cellular microviscosity, and it reduced the expression of drug efflux pumps, and that is why probably it is unable to form hyphae, and it shows increased sensitivity to the drugs. And again, it also showed synergism, as you can see here. Even at reduced concentration of silver nanoparticles and reduced concentration of the drug, you can see the cells are not visible here. So enhanced effect on cells when combined with other drugs, and reduction in dose of drugs ranged from 4 to 200 times, and it can be developed as a biofilm inhibitors. Then the next part is the water-soluble biocompatible multifunctional DNA dots. So this DNA dots were prepared by single-step process using salmon sperm DNA precursor. And this paper we published this year in Journal of Material Chemistry B. And so this is how, I'm not going into the details of the characterization. So we could find that we are able to make the DNA dots of 3 nanometer size, and it did not show any toxicity towards the microbial cells. And as you can see, why it did not show any toxicity? Because it could not produce the reactive oxygen species. So probably this is a positive control, hydrogen peroxide that we used, and even with a concentration of 8 milligram per ml, we did not find any production of ROS increased as compared to the control. And we used rhodamin 6G, which is a well-known fluorescent tracer dye, which enters the cells by passive diffusion and used to check different biological functionality, membrane transport process. So we found that we did time-dependent uptake of the DNA dots, and we found that it enters the cells by passive diffusion and accumulates in the cytoplasm, and the maximum accumulation is at 30 minutes. So this is when it accumulates maximum, and then gradually it attains equilibrium. And this is how the DNA dots within the cells, it looks like. It emits a blue fluorescence, and this is rhodamin 6G. And so these are the confocal microscopy images. And then we could find that DNA dots, when we mixed with hot solution of uh, DNA, we found that DNA dots acted as a cross-linker and formed a nanocomposite hydrogel at very low DNA concentration. And they were good tunable gels, and they were fluorescent gels. And these are the FESM images that you see with 20 micron and 10 micron scale bar resolution. And so this is the DNA dot hydrogel, which can be used, and it shows blue fluorescence under UV light. So we could make the DNA dots with potential for multifunctional applications. We used it for bioimaging, and the fluorescent hydrogel can be used for drug delivery. So we are further working on it. And the hydrogel, this work we did in collaboration, and it was also used for dopamine sensing. And the last part, which Anam already presented, so I would just briefly tell about that we tried to we synthesized actually fluorescent carbon dots, which were water soluble and non-toxic. And we are developing it as nano trackers for study of infectious diseases. So we could develop 2 nanometer size fluorescent carbon dots. And these are the different characterization techniques that we used. And we found that it is composed only of the carbon and oxygen, as the EDX shows. And so these are the different characterization techniques. And then Anam conjugated it with ergosterol, the membrane sterol, and she also conjugated it with drugs. So I'm only presenting here with CD and ergosterol. So this is how it formed, and this is how it looks like. So we checked with UV visible, with increase in the concentration of the ergosterol, there was increase in the absorption. And the band gap was found to be 4.05 electron volt. And whereas in the fluorescence spectra, as the ergosterol concentration increased, there was a decrease in the fluorescence with increase in ergosterol concentration, probably due to quenching. And SEM showed aggregation. And these are the different characterization techniques we showed to show that it is stable. And using the FTIR spectra data, we confirm that the conjugation has happened. So this is for the CD ergosterol conjugate. This is ergosterol, this is the CD. And so this is how we confirm that the conjugation has happened. And this is how the cellular internalization of the CD's ergosterol nanoconjugate looks like. Now, it is very interesting that wherever the ergosterol is localized, there all you will see the fluorescence. So you can see on the periphery, on the membrane, and inside wherever the ergosterol is localized. So Anam is right now doing using these conjugates to study the membrane dynamics, and also she is using for studying the drug uptake kinetics in the resistant isolates, and trying to see that how the drug uptake and the efflux changes in from resistant to clinical to resistant isolates. And she would be checking that how the hyphal morphogenesis occurs. So she would be using this CD ergosterol conjugate to check the hyphal morphogenesis. So we published these 13 papers in the last 19 and 20. So to conclude, nanomedicine is still in its infancy, and realization may take years, but still in the long run, it can be more economical, safe, and could greatly reduce the cost of medical procedures. And it is the future medicine. So I acknowledge my funding sources: ICMR, JNU UP2, JNU DST PERS, CSIR, and DBT, my collaborators, and my lab members. Thank you for your attention.
Thank you, ma'am. We have some questions. The first question is, now that the gram-negative bacteria have also started developing the antibiotic resistance, and with the current elongated drug development procedures, what are some ways that we can tackle this problem?
The combination therapy may be a good option to start with. And also repurposing drugs, because there are many drugs which are known to be useful for other diseases. So there are databases, and people are of course trying to repurpose the drugs which are known to be functional for some other diseases, and it might be good for some other diseases also. Like artemisinin is known for malaria, uh, cerebral malaria, but these days it has also shown functionality towards mycobacterium tuberculosis. So that ways, either repurposing the drugs or maybe combination therapy might work.
Okay, ma'am, thank you. The next question we have is, can we extrapolate the toxicity studies conducted on HeLa cells to all other cell types in the body? And does this toxicity of QDs differ in vivo?
It may differ a little bit in vivo. Uh, as far as other cell lines, it is the HeLa cell line toxicity test is a representative test, but of course, maybe we can check in couple of more cell lines which we are currently presently doing. And for in vivo work, of course, because in vitro condition and in vivo condition, there are a lot of other factors also acting and complicating the situation. So there the results may alter a little bit, because then the immunogenic factors also come into play.
Okay, so the next question we have is that apart from the material properties of silver, does the morphology of silver nanoparticles also play a role in its antimicrobial properties?
Uh, Pardon, can you repeat it once more?
Yes, ma'am, sure. Apart from the material properties of silver, does the morphology of silver nanoparticles also play a role in its antimicrobial properties?
Yes, yes, it is size dependent. So the smaller the size, the better will be the antimicrobial effect. But the size has to be tuned properly, because if it is too less in size, like especially for the metal nanoparticles, if it is less than 10 nanometers, then it is of the same size as the cell organelles. Then it is likely to interfere with the functioning of the cell organelles also. So there has to be a balance between the size. So too large size will not have good effect, and too small size will have toxic effect. So there has to be a balance in the size of the nanoparticles that are synthesized. So it is better
Uh,
to try to use particles which are more biocompatible and non-toxic.
Okay, ma'am. So I'd also like to ask that does the shape also matter? Like shape of the silver nanoparticles?
Yes, shape will of course matter, because then the surface area to the size ratio would change. So the more surface area to size ratio, the better is the activity.
Okay, ma'am.
And the
The next
permeability within
question
the
that
cell, the permeability within the cell also would differ depending upon the size.
okay. So the next question that we have is what is the composition of the DNA dots, and are they solely DNA or DNA linked with some other template?
It is not linked with any other template. So it is the simple DNA dots, and they are composed of the carbon and oxygen, whatever the simple DNA dots, and they are prepared by the one-step process from the salmon sperm DNA as precursors.
And
So
so
they are
how do
not
they
having any light.
okay. So like how do they differ from the regular DNA that we have?
That is as far as because if you change the size, they are dots, so they don't have, they are just 2 nanometer in the diameter, so they are just having almost negligible dimension. And the normal DNA would be a long elongated structure, the double helical structure. So this has been made from the single stranded DNA, uh, and then they have been converted through the chemical synthesis into dots which are the quantum dots, you can say. So they have the negligible dimension, and because of being in the dot form, lot of optical and physicochemical properties can be tuned, which you would not
Okay.
find in the normal DNA structure.
Okay, ma'am. So the next question is uh, that the DNA hydrogels that have been shown to stimulate the responsive behavior, how can such materials be used for development of sense act devices?
They can be used as a as a substrate, which can be used for binding with your specific antibodies and antigen reactions. So they can be used as a template, or as a basis for where you can bind your antigen and antibody.
Okay, ma'am.
For
The
the sensing
next question.
application. So
Okay.
we used it for dopamine sensing. So it was done by our collaborators. So I don't work on nanobiosensors.
Okay, okay, ma'am. So the next question that we have is it possible to convert any drug to a nanoscale? Or are there any certain parameters which we need to be taken care of or need to be checked?
Drugs can be converted to nanoscale, because see, curcumin is one example, which is a very common example. So curcumin, as you know, it has nanocurcumin has also been formed and synthesized or developed. And if you there are advantages of having the nano form, because then you can tune the functionality, and the bulk form of a drug and the nano formulation of a drug would differ in their functionalities and the properties also.